Okay, so your first lecture exam is on Thursday next week. That's 31st, January 31st. Right? If you see your schedule, lecture schedule, it is there, January 31st, next Thursday, okay? And the materials are from the beginning to where we stop next Tuesday. That's the last lecture before the exam. So where we will stop, I don't know yet where I will stop. So wherever I stop, that part will be covered. Okay? Thursday will be covered. Okay. So we have already talked about two chapters. One is an orientation of human body, or human body and orientation. We have talked about that, and we have talked about the chemical level of organization, right? And we did talk about the proteins, carbohydrates, fats, those. Okay. So today we will talk about the cellular level of organization. You know that the smallest level of organization of the body is the chemical level, and we have talked about atoms, elements, right? Those things. And one step higher level of organization is the cellular level. And cellular level of organization includes cells and the organs of the cells. So in today's lecture, first we'll see how to define a cell. Then we'll talk about different parts of a typical cell. Then we'll talk about the structure and functions of a cell membrane. Okay, we'll see the structure of a cell membrane and the functions of a cell membrane. Then we'll talk about the proteins in the cell membrane. If you see the structure of a cell membrane, you will see different types of protein structures are present in the cell membrane. So we'll see what are those protein structures, number one. Number two, we'll see what are the important functions that are performed by those protein structures. Okay, we'll see that. Then we'll talk about cell junctions. Many cells in our body are attached to each other. Those are the cell junctions. Those structures connect the cells to each other or attach the cells to each other. Those are the cell junctions. We'll see what are different types of cell junctions present in our body. Then we'll talk about transport across the cell membrane. Okay, now cell membrane is a semi-permeable membrane. Semi means partially permeable. That means a cell membrane will partially allow the chemicals to pass through it. Some chemicals will be able to pass through it, but not all. That's why it is called semi-permeable. It will selectively allow the chemicals to pass through it. It will block some, it will let some to pass through it. So we'll see how the chemicals are transported across the cell membrane. How the chemicals pass from one side to the other side to the cell membrane. Then we'll talk about tonicity. What does isotonic, hypotonic, or hypertonic mean? And then we'll see the effect of those three different types of fluid on a living cell membrane. If you put a living or alive cell membrane in isotonic, hypotonic, or hypertonic solution, how the cell will react, that we'll see. Okay, how the living cell will react in those three different conditions. So those are the things we will talk about today. First, how to define a cell. The cell is the smallest structural and functional living unit. Human body consists of trillions of cells. And trillions of cells, they together form the body. And cells are the structural and functional units. That means the smallest structure in our body that can perform its function individually. That is very important. That a cell can stay alive and perform its function individually. Example, how you know that a single cell can stay alive and perform its function individually? The best example is bacteria. You know, bacteria are single cell microorganisms, right? They can stay alive and they can perform their function. So, if you take a cell out from your body and keep it in a petri dish, put solution, glucose, nutrients, and oxygen, the cell will stay alive. And we do that. When we do research on cells, we take the cells out from the body and put in a dish where we give continuously supply oxygen, nutrients, and glucose. The cell will stay alive. Okay? So you know that individual cell can stay alive and perform its function. Right? That's why it is the smallest structural and functional unit of the body. Now, if you break the cell membrane, it will die. No way you will be able to keep it alive if you break the cell membrane, right? So if you go further small, it will not stay alive. So that's why cell is the smallest unit. Human body has 50 to 100 trillions of cells, and more than 200 different types of cells are found in our body. More than 200 different types of cells are found in our body. How they are different? They are different in shape, they are different in their size, they are different in their functions. Okay? So that's how the cells are different from one to another. And if you see the chemical structure of a cell, all cells composed chiefly of CH, N, and O. And we have seen that our body mass, 96% of the body mass is formed by carbon, hydrogen, nitrogen, and oxygen. So cells are formed by those four chemicals mostly. Okay, now we know that in our body more than 200 different types of cells are present. Here in this picture you can see a few of them, how they are different, how they function differently. I'll just quickly go over. So first one, upper left, those are fibroblasts. You see two fibroblasts, and fibroblasts are connective tissue cells. And the main function of fibroblasts is production of fibers. They produce fibers, connective tissue fibers. That's why you see around the fibroblast, some fibers are there. Next one is, these are erythrocytes. Erythrocytes are red blood cells in our body, red blood cells. Now, if you see the shape of erythrocytes, erythrocytes are round but flat, not like a ball, not like a round ball, round but flat, like a donut, right? And why the erythrocytes are flat? Because there is no nucleus inside. Okay, so that's why they are flat. But they have what? They have hemoglobin inside, and hemoglobin can carry or transport oxygen. So the main function of red blood cells is transportation of oxygen. So you see the shape is different than fibroblasts, the function is different than the fibroblasts, right? Epithelial cells. If you see the epithelial cells in our body, epithelial cells always stay connected to each other. they are not separated, they are attached strongly to each other. And epithelial cells form the body surfaces, like your skin has the epithelial cells, and they are connected to each other. Next, you see the muscle cells. This is a skeletal muscle cell, and these are smooth muscle cells. You see, even muscle cells are different from each other. Skeletal muscle cell is very long and multi-nucleated. Many nuclei are present inside. Smooth muscle cell is small and spindle shaped. That means middle part is wide, both ends are pointed, okay, and has a single nucleus inside. So just you see that how even muscle cells are separate, different from each other. One type of muscle cell is different from another type. Then the bottom left, this one is a fat cell. And fat cell is interesting because inside the fat cell it is mostly empty. Inside is mostly empty, and that's why the nucleus cannot stay in the center. It is present in the peripheral part attached to the cell membrane, because there is nothing to hold the cell in the middle of it. Okay, so that's how the fat cell looks like. We know that most of the cases the nucleus is present in the center of the cell, but this is different. Next one is a macrophage. If you see a macrophage is a large cell and has many processes or extensions from the membrane, cell membrane. You see many hands like a structure, hand-like structures are present, and those extensions help in phagocytosis. So the main function of a macrophage is phagocytosis. It can engulf the microorganisms, and those extensions help them to engulf the microorganisms or take the microorganisms inside their body. Yes.
Would you say hydrocytosis or fibro?
Phagocytosis.
Oh,
phagocytosis.
pH.
Yeah. pH. Yeah. Phagocytosis. They are eating. The right top one is a nerve cell or a neuron, and nerve cell has a cell body and a number of processes. So a nerve cell has a cell body and a number of processes, and these short processes, you see here, these short processes are called dendrites, and this long process is called the axon, and this is the cell body, right? So the nerve cell or neuron has dendrites and axon. Okay. So dendrites are short processes, axon usually one in a nerve cell, and long process. Now, the function of a nerve cell. We know that the nerves or neural tissue transports the signal by electrical means, right? Electrical signal transmission. When we have talked about the nervous system, I have mentioned that, right? Very fast electrical signal transmission. So what happens, the nerve cells, the dendrites of nerve cells will bring the electrical signal into the cell body, and then from the cell body the electrical signal will get out through the axon. So dendrites will bring the electrical signal in, and the axons will take the signal out. Axon will take the signal out from the cell body. Okay, so the function of a nerve cell is completely different than the functions of other cells we have talked about. Now the last one, you see, it's a very interesting shaped cell. That's a sperm, male sex cell, and it's like a small, tiny or baby fish. Right? It has a head a tail and a midpiece or body part. Why it has a tail? Because sperm needs to swim inside the body in the semen. So that's why its shape is like that, and its function is completely different than other cells. It is a sex cell and it fertilizes the egg. So just few examples, but we have more than 200 different types of cells in our body. Okay. Now we'll talk about the structure of a typical cell. You have studied cell in your high school, in your intro bio, in those classes. So I'm not going into detail and don't want to spend a lot of time to talk about the cells. All of you know. Just briefly, I will tell you something, and you need to remember those for you. Not going into detail because this is mostly in this class we talk about the anatomy and physiology, not those basic things. Okay. So anyway, so quickly I'll go over the structure of a typical cell. A typical human body cell has three basic parts. The cell membrane, also called plasma membrane, same thing, cell membrane or plasma membrane, that provides boundary. That means separates inside the cell from outside. And then if you see inside the cell, this is the cell, this is the cell membrane, and if you see inside the cell, you will find two things. A nucleus, which is called the control center, and outside of the nucleus, this part is called cytoplasm. Okay, so cell membrane that provides the boundary. Then inside the cell, you have two things: the nucleus and the cytoplasm. In between the nucleus and the cell membrane. Now, inside the cytoplasm, many cellular organelles are present. And those organelles, some of them have membranous boundary, and those are called membranous organelles, and some organelles don't have membranous boundary. Those are called non-membranous. Very simple, right? Membranous organelles and non-membranous organelles. Okay. Now, this is a nice, beautiful, colorful picture of a typical cell. And they have dissected the cell and have shown inside. But remember, where you see the real thing, it is way different than the picture. When you draw the picture, right, you can put everything there nicely, colorful by different colors, right? But the structure, if you see the real cell, it's not that colorful, not that beautiful. Okay, so just keep that in mind. This is a picture. Anyway, so it helps you to understand how the cell inside the cell looks like. So first, those three parts: cell membrane, nucleus is the control center, and cytoplasm. Now, in the cytoplasm, you have the organelles, membranous and non-membranous organelles. I'll just mention a few organelles and their important functions only, so you need to write it down or remember for the exam. Okay, so these are mitochondria, and mitochondria are membranous organelles, and the main function of mitochondria is, it is called the powerhouse. Mitochondria is called the powerhouse of the cell. Okay, powerhouse of the cell. Why mitochondria is called the powerhouse? Because mitochondria produces most of the ATPs, and ATPs provide energy. We know that, right? ATP is the source of energy. That's why mitochondria is the powerhouse. Energy. And now, if I ask you in the test, which one is the control center of the cell? Nucleus. Which one is the powerhouse of the cell? Mitochondria. Okay, so don't confuse. Okay, then another structure around the nucleus. This blue structure looks like many tubes are attached to each other. Many tube-like structures are attached to each other, and that structure is called the endoplasmic reticulum, or ER. Now, if you see the endoplasmic reticulum, it has two parts. In one part, in this part of ER, you see many tiny dots are attached. Those tiny structures are called ribosomes. Okay? Ribosomes are attached to the surface of a part of the endoplasmic reticulum. So, the surface of that part is rough because of ribosomes. So, that part of ER is called rough endoplasmic reticulum, or rough surfaced endoplasmic reticulum. Makes sense? Because many dots, ribosomes are attached, so the surface is not smooth. So it is the rough endoplasmic reticulum, or rough surface endoplasmic reticulum. The other part, if you see this part, does not have any ribosomes attached. So this part, the surface is smooth. That's why this is called the smooth surfaced endoplasmic reticulum, or smooth ER. Okay. Now, I'll just mention a couple of important functions of ER. First, rough endoplasmic reticulum. Number one, synthesis of proteins. This part is done by the ribosomes attached to the rough ER. So the ribosomes on rough ER are responsible for the synthesis of protein. And then after the protein is synthesized or produced by ribosomes here, the protein molecules are sent inside the tubes. And inside the tubes, folding of proteins take place. So folding of proteins. And also rough ER is responsible for transportation of proteins. Okay. So after the folding is complete, then the folded proteins will be sent to the Golgi apparatus. This structure, we'll talk about that. Just know that after the folding, the transportation means the proteins will be sent to the Golgi apparatus. So those are the functions of rough ER. Now, what are the functions of smooth ER? Smooth ER number one, synthesis of lipids or fats. Number two, metabolism of carbohydrates. Okay, metabolism of carbohydrates. And number three, detoxification of chemicals. Okay, so synthesis of lipids, metabolism of carbohydrates, and detoxification of chemicals. What is detoxification of chemicals? We take many chemicals inside our body. You know, you take medicines, right? Drugs. We eat the food. Foods are chemicals. And when we eat the foods or drugs, many harmful chemicals are there, right? So those must be detoxified. Those toxic chemicals must be neutralized, right? So that part, neutralization or detoxification, is done inside the smooth surface ER or endocrine. Okay, so that is a very important function of ER. Okay, now another structure, this green structure here, this is called Golgi apparatus. And Golgi apparatus looks like many tube-like structures are attached to each other, but those tubes have expanded ends. Both ends are expanded and they are piled together, okay, like this. And you know that rough ear will transport the protein, will send the protein to the Golgi apparatus. Okay, so protein will come into Golgi apparatus from ER, and then what the Golgi apparatus will do? Golgi apparatus will send the proteins to the ends of the tubes, expanded part of the tubes. So the protein molecules will accumulate here, and then what will happen? The expanded end will get detached from the tube and will form a cycle. A cycle. So a cycle means round membranous structures inside the cell, and they contain chemicals. So the end part, first the protein molecules will accumulate there and will get detached from the Golgi apparatus and will form the vesicle, and then the vesicles will move to the cell membrane and will rupture, and the chemicals or proteins will get out from the vesicles. And the process is called exocytosis, the rupture of the vesicles and the release of the chemicals or proteins from the vesicles to the outside of the cell. That process is called what? Exocytosis. Okay? So that's the function of the Golgi apparatus. So just remember those few structures and their functions. Now, we'll talk about the plasma membrane. Plasma membrane separates inside the cell from outside. That is number one. Number two, plasma membrane gives the cell its shape. So the shape of the cell is given by the plasma membrane. So those are two important functions of plasma membrane. Number one, it separates intercellular fluid from extracellular, that means inside the cell from outside. Number two, plasma membrane gives shape to the cell. Now, if you see the structure of the cell membrane or the plasma membrane, it is a bilayer of lipid and proteins. So lipids and proteins are present in the plasma membrane, and they form what? Bilayers. That means two layers. Two layers of lipids and proteins. And there is a theory of the structure of cell membrane that is called the fluid mosaic model. What is that? So cell membrane has bilayer, that means two layers. Okay, so two layers, and inside the lipids of cell membranes you will see the proteins. So these are proteins and lipids. Okay. Now, if you look the cell membrane from the top, if you look from the top, under the powerful microscope under a under heavy magnification, if you see the cell membrane from the top like this, it will look like, sometimes in the beautiful houses, you will see the floor has a lot of stones, right? Beautiful stones or marbles are used to make the floor. And it will look like that. It's like mosaic, a lot of beautiful stones. Those are the proteins in the white phospholipid or fat bilayer. So now that is the mosaic. Now number 2 is called fluid mosaic. What is that? The reason it is called fluid mosaic because those proteins are not fixed. This is very interesting. These proteins or marbles or rocks change their shape continuously. They change their shape. Make sense? So when you see under the microscope, you see beautiful like mosaic, and the rocks are changing their shape. So it is changing, dynamic. It's not fixed. The appearance is changing all the time. So getting changed all the time. So that's why it's called the structure of fluid mosaic model. Okay. Now what are the lipids in the cell membrane? Cell membrane has lipids and proteins, right? Those two things. So what are the lipids present in the cell membrane? The lipids are phospholipids, glycolipids, and cholesterol. Those 3 types of lipids are present in the cell membrane, the lipid part of the cell membrane. Okay. Now, phospholipids form bilayers, 2 layers in the cell membrane. And if you see a phospholipid molecule, this is a phospholipid molecule. It looks like this. Okay, and phospholipid molecules. And we know that phospholipids form bilayers. That means 2 layers. This is 1 layer. This is another layer, bilayer. And in the phospholipid bilayer, the protein structures are embedded, inserted inside the phospholipid bilayer. Okay, so like this. So I'm not good in drawing, okay? But not laugh. Okay, so you see, this is a phospholipid layer, this is a phospholipid layer. So phospholipid bilayer, and proteins are embedded or inserted. Okay. Now, if you see one phospholipid molecule, it has a head part and a tail part. Okay, and head part is called polar end, polar, and tail part is non-polar. And the polar part or head part of phospholipid loves water. That means hydrophilic. Philia means what? Attraction, right? Or love, or liking. So head part is also called hydrophilic. Philia. Philia means attraction or love or liking. So hydrophilic, hydro means water, hydrophilic part is the head part. And tail part hates water, doesn't like water. So this part is called hydrophobic. Phobia. Phobia means fear, scare, disliking. Okay? So hydrophobic part and hydrophilic part. Okay. Now, so these are the proteins inserted in the phospholipid bilayer. So there are 2 types of proteins present in the cell membrane. uh, one type of protein is deeply inserted in the phospholipid bilayer. Those are called integral proteins. If you see here, these proteins are deeply inserted in the phospholipid layer. These are heavily inserted. Okay, so these are called integral proteins. And some proteins are loosely attached to the outer part. This one. This is called peripheral protein. This is also loosely attached, not going inside the phospholipid bilayer. So this is the peripheral protein. So two types of proteins. One is inserted deeply inside the phospholipid, that is the integral. Now, those integral proteins, if you look, some are passing through the cell membrane all the way from one end to the other, like this one, passing all the way. Some are partially inserted, not completely inserted from one side to the other side throughout. So, but both are integral because they are deeply inserted. Okay. Now, if you look, this integral protein and this integral protein, there is an interesting difference. This integral protein is solid, but if you see this one, if you cut that integral protein, you'll see a channel or passage through it. Okay, so some integral proteins have channels through it and through them, and let the chemicals pass from one side of the cell membrane to the other side. Helps in transportation. Anyway, so just see the difference between the integral, say, two different types of integral proteins. Now, other structures. So this is phospholipid bilayer. These are the protein structure, and other structures are one is called glycolipid. This yellow structure is a lipid cholesterol, and this green structure is a glucose. So these are glucose. Green structures are glucose molecules. So when the lipid cholesterol is attached to the glucose, we say the name is glycolipid. Makes sense, right? Glyco means glucose and lipid is the cholesterol or fat, right? So that's why together the lipid and glucose we say glycolipid, right? So this is a glycolipid structure. Now here, this is protein, right? This is protein. So protein is attached to the glucose, or glucose is attached to the protein. So in this case, together this structure will be called as glycoprotein, because glyco is the glucose and protein is the protein. Okay, so glycoprotein. The protein is attached to the glucose. So those are some cholesterols or lipids are not attached to the glucose. So those are free cholesterols. So you see that's the structure of a cell membrane. Okay. Functions of integral proteins. I told you that those protein structures in the cell membrane perform a number of important functions. Okay. So we'll see what are the important functions of integral proteins and what are the important functions of peripheral proteins. Integral proteins help in transport, that means transportation. Many integral proteins work as receptors, and some integral proteins act as carrier. They can carry the molecules, and some integral proteins form channels, have two channels or passes through it. Okay, so transport. So you see here two integral proteins, and we know that the phospholipid, this layer, since this layer, phospholipid bilayer, is mainly formed by lipids, phospholipids, so water cannot easily pass through the phospholipid bilayer. The problem is water is repelled by fat, right, or lipids. You know that lipids don't mix with water easily, right? So phospholipid bilayer will not let the water molecules to pass through it easily. That's simple. So what happens? But water must go inside the cell, right? So these proteins help in the transportation of water molecules or other water soluble molecules through it. And you see two integral proteins here. One has the channel which is always open, stays open all the time. Okay, so that will let the molecules, those molecules have smaller size than the channel, size of the diameter of the channel. They will easily pass through, right? If the size of the molecule is smaller than the diameter of the passage or channel, they will easily pass through it. And those channels stay open all the time. Okay, now in this protein, integral protein, the channel remains closed most of the time, and to open those channels you need to provide energy to open the door. It's like door, right? If the door is closed, then you need to push it. You need to utilize energy, right, to open. So two types of proteins you see. One has opening all the time, another is closed, the door is closed, but it can be opened. To open that, we need to use ATPs. Our body needs to use ATPs to open it. Okay? So that's how the integral proteins help in transportation. So just remember, transport is one important function. Many integral proteins work as receptors. Very important function. See what happens. What does mean integral proteins work as receptors? If this is the cell, and so this is the cell membrane, and this is the integral protein on the cell membrane, okay, in the cell membrane. Now, when you take a medicine, drug, it's chemical, right? And those chemical molecules must work on the cells of your body. So what happens? The chemicals that will work on the cells will get attached to the integral proteins. Okay? And when that attachment will occur perfectly, then what will happen? The signal will go to inside of the cell, will go inside the cell. And that signal will start the intercellular machineries to start their functions. So now, these integral proteins, those work as receptors. If we look under the microscope, they have binding sites. The binding sites have different shapes. This is another integral protein. This is the binding site. Okay? So you see that we have thousands of different binding sites on the integral proteins, and they look different from each other. Now, if you take a medicine, the molecule is round. The shape of the molecule is round. Then this one will perfectly bind with which one? 3. Very good. This one, right? Because the shape, you see here, the binding site is here, is like that. It will be able to hold the round molecule. Okay, this is perfect. If it goes here, the binding will not be perfect. Make sense? So it's like you put the key in your car, right? And you have many keys, but you need the perfect one to start the car. So when that binding will be perfect, then the signal will go to inside the cell to start the engine. Okay, and then cell will perform the function. Now, if the shape is triangular, the molecule you are taking, that will perfectly fit here. Okay, so it's like key lock system. When you come here, you wait outside, right? Because you don't have the key. You have many keys, but then the key to open this door, you don't have that perfect key, right? So I have that key, so I can open the door. So that's why this is very important. See that these chemicals are called signal molecules. Signal molecules, okay? And these are the receptor proteins. So signal molecules must bind perfectly to the receptors. Okay, that is very important. And just think that you take different types of medicines, right? And the molecules of those medicines are different from each other. And when you take a medicine that should work on your kidneys, the manufacturers, the pharmaceuticals companies, they make the molecules, they know the receptor, the shape of the receptors on your kidney cells. Okay? So they make the molecules in a way that will bind perfectly on the kidney cells, proteins, and will work on the kidneys. If it goes to the liver or brain, it will not be able to get that. Make sense? So it will not work on those other organs. That's why you need to know the shape of the receptors, binding sites of integral proteins. Attachment to cytoskeleton and extracellular matrix. Just know that this is the integral protein, and some rod-like or tube-like structures are present both inside and outside of the cells. Those are called microtubules and microfilaments. I don't know if you have heard those. Inside the cell and outside the cell, we have microscopic tube-like and rod-like structures. Tube means they have opening inside, right? And rod means solid. So rod-like and tube-like structures we have both in outside and inside the cell. Those are called tube-like structures are called microtubules, and rod-like structures, solid structures are called microfilaments. And those structures stabilize the cell membrane. Otherwise, cell membrane will move easily. So to stabilize the cell membrane, those structures are important. Now, what happens, those tubes or rod-like structures, they get attached to the integral proteins, okay, both inside and outside, and hold the cell membrane stable. Okay, that is another function of integral proteins. It's like when you go for camping, right? When you go for camping, you take the tent, right? And you also, to put the tent strong, you need to use what? The poles, right? Use the poles. So it's like that. The cell membrane is the tent, and you use the poles to hold it or make it strong. Okay. So integral proteins help in that by providing attachment to the microfilaments and microtubules. Some integral proteins work as enzymes. How they work as enzymes? What's the function of an enzyme? The function is it helps in chemical reactions. So they have shown here that the two integral proteins side by side, but they are working as two different enzymes. The first one is converting this chemical molecule to this, and second one is converting this molecule to this. Just very simple way they have shown you that the chemical reactions are helped by the enzymes, and some integral proteins help in that, help in chemical reactions. That means they work as enzymes. Okay, so those are the functions of the integral proteins. Now we talk about the functions of peripheral proteins. Peripheral proteins perform three functions. Intercellular joinings. The cells are connected to each other by intercellular junctions or joinings. Same thing, junctions or joinings. Inter means in between, right? So intercellular means that in between the cells, those junctions or joinings help to connect the cells or hold the cells to each other. Cell-to-cell recognition. Another very important function of peripheral protein. What is that? You know, if you need tissue in your body from another person because you have lost some tissue in your body, then what we do transplantation, right? Transplantation. So, in most of the cases, if I give you tissue, my tissue to your body will do what? Reject it. We know that. Most of the cases, our body will reject it. And if I give tissue from another location of your body to your body, then it will take it, right? Accept it. So, what it tells it? It tells that your body cells can recognize, right? Which cells are from your body, which cells are from another body. Okay? So that recognition is extremely important. Okay? In tissue or cell transplantation or body part transplantation. So that recognition is given by the peripheral proteins. Okay? The peripheral proteins help in that. We know that integral proteins help act as enzymes. Also, some peripheral proteins act as enzymes. Okay? So those are the three important functions of peripheral proteins. Just remember, if I ask you that cell-to-cell recognition is a function of integral or peripheral protein, it is a function of peripheral protein. Okay? Remember that. If I ask you that intercellular junctions or joinings are formed by what type of proteins? Peripheral protein. Okay? Transportation is helped by which proteins? Transportation. Integral proteins. Okay? Integral proteins. Which proteins work as receptors? Integral proteins, right? Integral proteins. So remember that. Just remember important functions. Now we'll talk about the intercellular joinings or junctions. We know that peripheral proteins form intercellular junctions or joinings. So three types of junctions are very commonly seen in our body. What are those? Tight junctions, desmosomes, and gap junctions. Those are three types of intercellular junctions or joinings are seen very commonly. Okay? Tight junctions prevent fluids and most molecules from moving between the cells. So, if this is one cell and this is another cell, the cells are connected by junctions, and tight junctions are the junctions. Those are heavily sealed. So, the molecules of fluid will not be able to easily pass through the tight junctions. Okay? Will not be able to move from one cell to another cell. Desmosomes anchor the cells very strongly. This is the strongest junction among those three. Very strongly hold the cells together. We'll see in picture how they do that. Gap junctions. By their name, you can tell that gap junctions have many gaps or openings. So they have many channels through them, so the fluid or molecules can easily pass from one cell to the next. they have many openings or gaps inside. So tight junctions will not let the molecules or fluid easily pass through it. Gap junctions are opposite. They will let the fluid or molecules easily pass through it. And desmosomes hold the cells together very strongly. So we see the pictures of those three types of cell, junctions. First, this is a tight junction. Okay? And if you see the structure of a tight junction under the microscope, you see two cell members, adjacent cell members, are attached to each other strongly by like stitching the cell members together, stitching them together. So many interlocking junctional proteins stitch them together. So this is the tight junction. It will not let the fluids or molecules pass through. And these are desmosomes. Okay, so we'll see the desmosomes probably next picture. Let's see, it's there. Yeah. So if you see a desmosome, this is one cell membrane, this is another cell membrane, and in the inside part of the cell membrane, this structure is called plaque. It's like a disc. If this is the cell membrane, a solid disc is attached to the inner part of the cell membrane, round disc. Okay? And those are called plaques. And from the plaques, you see linker glycoproteins come out, and those linker glycoproteins from both sides, from both discs or plaques, linker glycoproteins will get out, and those linker glycoproteins, if you see the structure, they're like this. These are linker glycoproteins coming from one side. These are linker glycoproteins coming from the other cell membrane, other disc. Okay? And they get attached to each other like this. You use the zipper in your clothes, right? That you know when you close it, how strong, right? It's very strong, you won't be able to separate. So similarly, glycoproteins, linker glycoproteins, they hold each other like this very strongly. You won't be able to separate. Okay? That's why the cell membranes stay together very strongly. Here, just remember one thing. I may ask that those linker glycoproteins are also called cadheres. So cadheres are linker glycoproteins in desmosomes. Okay, remember that name. I may ask, not here. Okay, gap junctions. If you see the gap junctions, as I have already mentioned, that many pores or holes are present. So the molecules or fluid will be able to pass through those channels. Okay? So inside the gap junction, you will see many true passages. So the fluid will be able to pass, or molecules will be able to pass from one cell to the next. Okay, so those are three different types of important types of intercellular junctions. Yes.
So are all of these junctions in one cell to or two cells? Or there like there'll be some in one and others?
Yes.
Because your picture shows like it's,
Yes.
they're all three exist in one cell.
Yeah, that's why the picture is to make it clear to you. We're using colorful.
But they don't all
pictures.
exist
That's why
in one cell.
exactly.
They're just
It is very
they'll have
rare.
one type in one.
One or two types. Most of like in your cardiac muscle, heart muscle, you have desmosomes and gap junctions. Those two. Okay, no tight junctions. Most of the cases, the epithelial cells in our body, and Do you remember that I showed you different types of cells in our body? Epithelial cells are attached to each other, I told. So these junctions are mostly found in epithelial tissues where the cells must be attached to each other. And like one or two, not all three are present in one cell, one tissue. That's rare. I don't know any. But in your heart muscle, you will find two, desmosome and the gap junction.
Will you usually find tight junctions and gap junctions in the same?
Tight and gap could be same in epithelial tissue, yes, you can find. Now, the heart muscle has both gap junctions and desmosomes. The reason is that heart muscle must contract forcefully at the same time, right? To pump the blood forcefully, pump the blood out. Now, the desmosomes hold the cells very strongly to each other, right? So see here, if this is one heart muscle fiber, this is another, they are connected by desmosomes strongly, right? So when this will contract, it must pull the other one. No way other one will stay relaxed and will say, I won't contract. It must. So all the heart muscle cells are connected by desmosomes, right? So when they will contract all, whole thing will get shot at the same time. So all of them are attached to each other. That is one thing. Another, the fluid between the cells must be exchanged very fast in the heart muscle. That's how the heart muscle cells talk to each other. Like if the door is open between two rooms, you can easily communicate, right? Talk to each other. But if the doors are closed, what's going on in one cell or one room, the other room will not know, right? So that's why many gap junctions are also present in the heart muscle, and the cells, if anything occurs in one cell, next cell will easily know because the doors are open, right? Gap junctions are there. So those two junctions are very important in the heart muscle to perform its function. And that's why it is called that heart muscle works as a syncytium. Syncytium means there are thousands of cells, right? But they are all working as a single unit because the doors are open. So you can say that this is one big unit, right? They are not separated. That is called syncytium or a unit, a single cell. In your skin, the cells are connected by desmosomes. Makes sense, right? You know that the skin protects the body, number one. And when the skin gets dry, you can feel the skin. A chunk of the skin you can feel, right? You can't separate the skin cells. And particularly if you see the snakes, when they change the skin, you have seen probably, the snakes, they change the skin, the whole chunk of the skin comes out, right? And why? The cells are connected heavily, strongly by desmosomes. So in the skin, you see plenty of desmosomes. They hold the cells together. That's why big part of the skin comes out together. The cells come out together. Okay. So now we talk about the transport across the cell membrane. You know that cell membranes are semi-permeable membranes. That means partially permeable. Will selectively allow some molecules to pass through it, not all. So what are the transports occur through the cell membrane? We can divide the transports into two types: passive processes or passive transports, active processes or active transport. Passive transports don't need energy or utilization of ATPs. We know that ATPs provide energy, right? So in passive transports, no ATPs are required. No energy is required. In active processes, energy or ATP is required. Okay, so we need the utilization of ATPs in active transports. That's why they are called active. We need energy. Now, if I ask you, that which one can only occur in the living body, the active one, right? Because we know that to produce ATP, we need what? Mitochondria, right? Mitochondria is the powerhouse. So ATPs are produced in the mitochondria. And if the body is not alive, then mitochondria will not be able to produce, metabolism will not occur, ATP will not be produced, right? So only active transports occur only in the living bodies or living cells, living cell members. Okay, now both the active and passive transports have different types. What are the different types of passive transports? Simple diffusion, carrier mediated facilitated diffusion, channel mediated facilitated diffusion, and osmosis. So those are 4 different types of passive processes or passive transport. Simple diffusion. Simple diffusion is the process by which the molecules move from the high concentration to the low concentration. Okay, so that is simple diffusion along the concentration gradient. If this is the cell membrane, okay, and in this side you have a glucose solution, high concentration glucose solution, and in this side you have low concentration glucose solution. Okay, so the pressure is high in this side. Okay, so the glucose molecules will move from high to low. So who will move? Glucose molecules will move from high to low, and this is the simple diffusion. Doesn't need any energy. Now remember, this is glucose solution. So what are the things you have here? You have water and glucose. That's how we make the glucose solution, right? So water, in this case, water is solute or solvent? Water is solvent, right? And water is the solvent. Who is the solute? Glucose. Glucose is the solute. In simple diffusion, who will move from high to low? The solute. So the solute will move from high to low. The purpose, remember that. Always you need to think why it is moving. Concentration is high in this side, right? And low in this side. Now, if glucose molecules move from this side to this side, then gradually the concentration in this side will go up. Make sense? And in this side it will go down. So the purpose is bringing equilibrium, same concentration in both sides. So to do that from this side, just now think that to do that from this side, who should move? The glucose, right? Glucose should move from this side to this side, and that is what? Simple diffusion. Now, at the same time, what happens? The water molecules will move from this side to this side. The purpose is same, right? Because water is less in this side, more in this side, right? The concentration is less in this side, that means water is more, right? So the water molecules will move from this side to this side. And the purpose is same, to bring equilibrium. So water will move from this side from low to high, and the glucose will move from high to low. So when glucose will move from high to low, that is solute is moving from high to low, that is called what? Simple diffusion, right? The solute is moving from high to low. And water is moving from low to high, and this is called osmosis. Okay, osmosis. So you see, both simple diffusion and osmosis will occur at the same time through the cell membrane. The purpose is bringing equilibrium. When water will move from here to here, the concentration will go down, when glucose will move from here to here, here concentration will go up, concentration on this side will go down. And eventually, on both sides, the concentration will be same. So now you know what is simple diffusion, what is osmosis. Okay? Is it clear? Difficult? No? Okay.
Did you say it moves with the gradient or against?
In case of simple diffusion, the solute will move towards the concentration gradient. That means from high to low.
Right.
Okay? And in case of osmosis, it's going in opposite direction.
So that's moving against the
Against
gradient.
the gradient. Now, in the test, if I ask you that in which one, in simple diffusion or osmosis, the solutes will move from high to low. Solutes.
Oh,
Simple diffusion. Is it clear to everybody? The solutes. In this case, the solute is glucose, right? And solvent is water. So, in osmosis, the solvent will move from low to high. Is it clear to everybody? Okay. Good. Say loud. Yes. If not, then ask me again. Okay? Don't feel shy. Yes. Good.
Okay, so you said there was more water on that side, right? 'Cause
Yes.
Like, so it's still moving with the gradient, right? Because it's moving to low.
In that way, you can say that,
Okay.
yeah, in that case, you can say that if you just think the water, it's moving towards the gradient.
Okay.
So how you say that? Okay. So carrier mediated facilitated diffusion and channel mediated facilitated diffusion. We'll see what are those. Okay. Some integral proteins work as carrier. How they work as carrier? If you see here in this picture, inside this integral protein there is a binding site. So this integral protein can change the shape. This is very interesting. Can change its shape, and when it will try to transport any molecule, what it will do, it will open the upper part and will let the molecule get in. So this part will be open and the molecule will get in and will get attached here. If they didn't show here, it will get attached here, and then what will happen? This integral protein will, this upper end will get closed and lower end will open like this. Okay? So it's taking the molecule inside, molecule will get attached to the protein, hold it there, then upper part will get closed and lower part, other end will open like this, and will let the molecule get in. Okay? So
Are those sites specific to the
yes, yes, specific to molecule. Okay? Shape of the molecule.
binding proteins?
Yeah. So this is called carrier mediated diffusion, facilitated diffusion. Channel mediated facilitated diffusion is very simple. As I showed you before, that some integral proteins have channels, and those integral proteins, those channels will always stay open, right? So smaller, only the important thing here that the molecules that will pass through it must have smaller diameter than the diameter of the channels. Make sense? So if the molecules are smaller than the size of the channel, they will pass through. That's the channel mediated facilitated diffusion. We have talked about the osmosis. We have talked about the simple diffusion. Just ignore these here. A nice experiment you can do in the lab. You see, this is an U-shaped glass tube, and you put a membrane. That membrane has large holes in it, and if you separate two different concentration solution here, this side has concentration is higher, this side, the concentration is lower. Okay, just think glucose solution that I have shown you. So in this side, these are the glucose molecules, and this is water. So more glucose molecules here, that means the concentration of solution is high here. In this side, water is more, glucose is less. That means the concentration is low, right? Now, since this membrane has large holes bigger than the size of the solutes, what will happen? The solutes will move from which direction the solutes will move? From high to low. Very good. And that is simple diffusion, right? The solutes will move from high to low. That is simple diffusion. So solutes will move to this side. And at the same time, the water will move from this side to this side. Make sense? Because concentration is high in this side, water is less, so water will move to this side. Always think that what's the purpose? Purpose is what? Equilibrium, bringing equilibrium. How you can bring equilibrium? The pressure equal, moving some solute from this side to this side, and moving some water from this side to this side, right? That is simple. So, when the solutes are moving, that is the simple diffusion. When the water is moving, that's the osmosis. So, both will occur in this case, and eventually, both sides will have same concentration of solutions. Okay? So that is one experiment. Now, if you put a membrane that has the poles smaller than the size of the solutes of glucose here, what will happen? Who will move now? Only water will move, right? Because the water molecule is smaller than this, but the solutes will not move. So in this case, since the concentration is high this side, the water molecules will move. The purpose is bringing same concentration, not the volume concentration, remember? So the water will keep moving until the concentration comes to equal. Although the volume has increased, but the concentration in this side and this side is same. And this is an experiment where you can see the osmosis, right? Osmosis, only the water or solvent is moving from one side to another side. Okay, now we'll talk about the last thing a little bit over tonicity. Tonicity is a relative concentration. So tonicity is what? A relative concentration. That means, relative means it's not fixed. Like if you see two guys are standing side by side, right? One is 4 feet tall, another guy is 6 feet. Now you will say that 6 feet guy is taller, right? And 4 feet guy is smaller. Now if you put 8 feet guy here, then 6 feet is smaller, 8 feet is taller, right? You see, you can change the term because of relative comparison. Okay? So remember that. So tonicity is not fixed, it's a relative concentration. Now, if this is a cell and inside the cell, the concentration of fluid is 5 milliosmol. Milliosmol is the unit of concentration. So 5, and outside fluid has 10 milliosmol. So in this case, we'll say outside is hypertonic and inside is what? Hypo. Hypo means less. Hypotonic. Okay. So outside fluid is hypertonic compared to inside, and inside is hypotonic compared to outside. Now, if I change this to 15, which one is hyper? Inside is hyper, right? And outside is hypo. So you see that by changing one, you can change the other. So it's not fixed. Now, if I change this one to 10, So both inside and outside have same, right, concentration. So this is called isotonic. Okay. Now, if you put a living cell in a solution, this is the cell, you drop a living cell in a solution that is isotonic. That means the concentration is same as inside the cell. In this case, the cell will not change the shape. No fluid will go in, no fluid will get out. Makes sense, because no osmosis will occur because the concentration is same, right? No osmosis will take place. Makes sense? So nothing will get in, nothing will get out. The shape of the cell will remain same. Now, if you put the cell in a solution that has this is hypertonic, right, compared to the cell. So if you put the cell in a hypertonic solution, concentration is high here, concentration is low here. Now you tell me, in osmosis, which way the water will move? Water will move from inside to outside. Very good, because water will move from low to high. And in this case, the size of the cell will get smaller, shrink. Does it make sense? So water will get out from the cell. Now, if you put the cell in hypotonic solution, in this case, water will get in. You go from low to high, right? So the cell will get bigger, swelling, and may burst, right? If more and more water goes in, the cell may burst. So the cell will get bigger and may eventually burst. Okay. So now, if I ask you, if you become a nurse and I ask you to push some saline inside the patient's body, you will pick isotonic, hypotonic, or hypertonic. Isotonic. Make sense? Because you don't want to bother the cells. You don't want to bother the blood cells in your blood, right? If you put isotonic, the shape will not change. If you put hypertonic, then the cell will shrink, right? If you put hypotonic, the cell will get bigger. We don't want, that's why we always use the isotonic solution.
Well, like if you're dehydrated, then your cells typically would be, they would shrink, right?
When you get dehydrated, the water part is less in your blood. Yes. So what will happen from the cell? The water may come out. That's, yeah, that can happen.
So then when you give like an IV or whatever, that's your.
Yeah. Yeah. In that case, your cells will get a little bit bigger because it's getting the water right from outside. Will get a little bit bigger. But always remember that when you are dehydrated, that doesn't mean that your bladder doesn't have water. Okay? So usually the cells will not change their shape unless significant amount of change occurs inside our body. That is the homeostasis. Our body and the cells of the body try to stay as they are. Okay? So and yeah. So when we get the cell in, we get usually 1 liter or 2 liters, right? So but still it is good not to give the isotonic or hypotonic or hypertonic, not to bother the function of the cell. It will not kill the cells, but if the cell changes the size or shape, that may bother the functions inside the cell. Okay, so that could be a problem. Okay, do you have any other questions? No? Okay.